In this lesson, we'll be looking at how different eukaryotic cells express different genes at different times, despite having exactly the same genome. In a previous lesson, we looked at how prokaryotic cells can influence gene expression at different times of the expression pathway. In eukaryotes, there's much more complexity to how genes are used to create protein, and so there are many more locations in this pathway for the cell to exert control. Within the nucleus of the cell, the packaging of chromosomes can greatly influence how frequently the DNA is transcribed. Once it's been transcribed, various ways of processing the transcript can lead to different mRNA molecules that are exported from the nucleus to the cytoplasm. Once the mRNA has been exported to the cytoplasm, the cell controls how quickly it's degraded and therefore how much it's translated. Polypeptides, once formed, may become proteins, but require several steps in order to complete the transition from polypeptide to functional protein. The cell may degrade the protein quickly or slowly and may control when and where the protein is activated. One of the most important determinants of how a gene is transcribed is how it's packaged. In the nucleus of the cell, the DNA is packaged by wrapping around proteins called histones. In fact, the DNA wraps around groups of 8 histone proteins that are collectively known as a nucleosome. The nucleosomes, in turn, may bundle together tightly, especially for meiosis, when the chromosome is condensing. However, during interphase, the chromatin structure is much looser. This looseness allows space for molecules such as RNA polymerase to access the DNA. If the DNA is wound tightly, then it's difficult for the transcription complex, including polymerases, to gain access to the DNA. Therefore, in order to make a copy as mRNA, the chromatin needs to be loosely structured, with space between each of the nucleosomes. This is achieved by the addition of acetyl groups onto the tails of histones. These acetyl groups force the DNA to be more open and therefore to be transcribed more. The flip side of this is DNA methylation. When DNA is methylated, a methyl group is added to certain nitrogenous bases, and this causes them to wrap around histones more tightly, decreasing access of the transcription machinery. Without access, the transcription machinery fails for that region of DNA, and transcription of that region is reduced. The most extreme form of this comes in the inactivated X chromosome in mammalian females. Remember that they only have one active X chromosome per cell, and the other one has been completely deactivated. This deactivation comes about through methylation, which tightens the chromatin structure, preventing any access of DNA polymerase and the other molecules necessary for transcription. In a much less extreme form, certain genes may be methylated, and these methylation patterns can actually be passed on from a cell to its daughter cells. Genes that may be turned off in one cell type of a body, for example, a liver cell, will also need to be turned on in the daughter cells of that liver cells, which are themselves liver cells. Therefore, a cell doesn't have to be reprogrammed every time it divides. The exception to this is that these markers are mostly removed during gamete formation. This makes sense because the gamete needs to be able to differentiate into a wide variety of cells. Okay, so we've seen how the very structure of DNA and how it's packaged can affect how often a region gets transcribed. Now, let's look at the next step, transcription. Remember that in order for a gene to be transcribed, there are many proteins that need to bind to the DNA. One of the most important, of course, is the polymerase, but other proteins are also needed to guide the polymerase. These other proteins are generally known as transcription factors, and they generally help in getting transcription started. They bind to a region of the DNA called the promoter that's upstream of the gene. There are a handful of transcription factors that are able to bind at all or most genes and aid in transcription of any of those genes. These transcription factors are called general transcription factors, and they typically bind at or near the promoter sequence. By themselves, they're enough to get some low level of transcription started. However, they usually aren't enough to produce large amounts of mRNA transcript. Located upstream of the promoter are proximal control elements, and much further upstream are distal control elements, often called enhancers. These control elements and enhancers greatly increase the rate of transcription because they tend to bind specific transcription factors. Specific transcription factors are proteins that are specific to a particular gene. Unlike the general transcription factors, specific transcription factors may only bind to and enhance transcription at a single or sometimes just a few genes. It's thought that enhancers, which are far upstream from the promoter, may help in transcription by bringing specific transcription factors to the start site of transcription. Here, you can see that the assembly of the transcription initiation complex is aided by the presence of these enhancers. The enhancer brings together a number of proteins that, working together, enhance transcription frequency. You can see from this model that the distal control elements are expected to be quite far from the promoter. In fact, they don't need to be upstream of the gene at all. They may be downstream of the entire gene, or they may be in an intron. The important thing is that they bring together different proteins into the correct place so that transcription factors, both general and specific, are able to increase transcription rates. One strange thing that scientists learned was that despite having thousands and thousands of genes, there's a very limited number of specific transcription factors, even though the transcription factors seem to be very specific for a particular gene or small number of genes. The way this works is that different genes in different cells will have specific combinations of the dozen or so specific transcription factors. Therefore, in order to initiate transcription, the particular combination of transcription factors must be present. In this example here, two cells from the same individual, and therefore having the same genome, express different genes during their life. Both of these genes require the gray transcription factor. The albumin gene on top requires red, gray, and yellow transcription factors, and the crystalline gene below requires brown, gray, and pink transcription factors. Although the gray transcription factor is common to both, you can see that the exact combination required is very particular, and transcription won't start at an appreciable rate until all of those transcription factors are in place. In the lens cell, on the right, the cell doesn't produce the same transcription factors needed to start transcription of the albumin gene. However, it does produce the necessary factors to get the crystalline gene transcribed. So instead of requiring a particular transcription factor, genes generally require specific combinations of those transcription factors. In contrast to this, sometimes there's a need to coordinate the transcription of many genes which may be located on different chromosomes. This may happen when the body needs to generate a variety of responses to a single signal. In this case, a number of genes may have the same or very similar enhancers. You should be able to contrast this with the operons in bacteria. Remember that with operons, the related genes were all located together on the chromosome and had a single promoter. In eukaryotes, however, the genes may be widely dispersed through the genome, and therefore each will have its own control elements. However, these control elements may be similar or the same as one another. Recent research has increasingly found that within the nucleus, each chromosome has a particular region in which it's located. Additionally, particular parts of the nucleus serve as transcription factories with concentrated reagents necessary for transcription. These tend to be located toward the middle of the nucleus, and so genes that are frequently transcribed are also located in that region, and genes that are less frequently transcribed are on the periphery of the nucleus. Remember that in eukaryotes, once transcription has happened, the primary mRNA transcript needs to be processed in a number of steps before it can be exported into the cytoplasm for translation. One of these steps involves splicing out the introns and retaining only the exons. This can be another point of control for differential gene expression, as some genes may splice together different sets of exons compared to other cells. In fact, most genes are capable of being alternatively spliced, and this is currently thought to be the primary reason why there doesn't seem to be a relationship between the complexity of eukaryotes and the number of genes. In other words, a complex organism is not complex because it has more genes, but maybe because it has more variety of alternative splicing, allowing the formation of more diverse proteins. Finally, once the mRNA has reached the cytoplasm, there are still a number of ways in which the cell can exert influence over protein production. The first involves how long the mRNA remains intact in the cytoplasm. Messenger RNA is inherently unstable, and some strands will only last for a few minutes. If the mRNA is short-lived, then the cell will need to produce more if it wants to produce more protein. Other mRNAs are relatively stable, such as the hemoglobin mRNAs. In order to start translation, a number of proteins are needed in addition to the ribosome. The cell is able to control which of these proteins may be available and may even be able to control translation universally by activating or deactivating necessary proteins. Some cells are held in stasis and don't grow until triggered by particular events. An example of this is in eggs, where translation is triggered universally shortly after fertilization. The last stages of generating proteins involve their activation. Remember that proteins often need to be activated through chemical modifications that make them fully functional. These may include additional proteins or the addition of phosphate groups. By controlling these modifications, the cell can control how many protein molecules come online at a given time. Finally, the length of time that a protein is active can also be controlled by the cell. The cell may tag some proteins with another protein called ubiquitin, which instructs yet other protein complexes to degrade the marked proteins. In recent years, biologists have increasingly found that non-coding RNAs play an important role in gene expression. The first way they do this is through RNA interference. In RNA interference, small pieces of RNA bind to a protein, often a protein called RISC. When the RNA is bound to RISC, it's able to recognize complementary sequences of mRNA. Once a complementary sequence has been located, the RISC protein will cut the mRNA or otherwise prevent it from being translated into protein. In this way, RNA interference either degrades mRNA or prevents it from being translated, but in either case, it interferes with translation. RNA interference can be carried out either by microRNAs, miRNAs, or by small interfering RNAs, siRNAs. The difference between these two is subtle, and researchers have increasingly been combining these into a single RNAi, or RNA interference category. Finally, evidence is mounting that non-coding RNA also plays a role in the structure of chromosomes and how tightly bound the chromatin is. Of course, the tightness of the chromatin affects transcription because tightly bound chromatin does not permit polymerase and other protein transcription elements to gain access to the DNA. Non-coding RNA, including long non-coding RNAs or lncRNAs, seem to be able to recruit enzymes that modify the chromatin, causing it to bind up more tightly. This is particularly noticed in the centromeres and on the X chromosome when it's turned into a bar body through transcripts of the exist gene. We've previously looked at what causes cancer. In general, we can say that cancer is caused by genetic changes that result in a loss of control of the cell cycle. As a result of these changes, the cell progresses around the cycle again and again without being inhibited by internal or external signals telling it to stop. Genetic changes that accumulate during this time are also passed on to the offspring of a cancerous cell, causing that cell to also become cancerous. Let's look now at a bit more detail of how the cell cycle control is maintained and what goes wrong in cancer. There are two important types of genes that we think about with cancer. The first type is called an oncogene. In fact, in normal, healthy cells, these genes are called proto-oncogenes. In general, these genes give signals for the cell cycle to progress. You can think of this like the accelerator on a car. A normally functioning proto-oncogene allows the cell to progress around the cell cycle as appropriate. However, if the proto-oncogene becomes mutated in a somatic or non-germline cell, it can cause the cell cycle to progress more rapidly, like the accelerator being stuck to the floor. Let's look at how a gene that normally helps progress the cell through the cell cycle becomes an oncogene, in which the cell cycle progresses in an uncontrolled way. This can happen in one of three ways. First, a proto-oncogene may be transposed or translocated to another part of the genome. When this happens, it may settle into a place with different regulatory elements. These regulatory elements may cause increased expression of that gene through any of the mechanisms that we previously talked about, such as increased transcription. When this happens, the protein products of the gene are more numerous and help to advance the cell cycle faster than normal. Secondly, a proto-oncogene may be duplicated. When this happens, each of the genes will produce mRNA, which then go on to produce protein. As a result, more mRNA and more protein is produced and cause rapid progression around the cell cycle. Finally, there may be a mutation either in the control element or within the gene itself. When such a mutation is in the control element, more gene products or proteins are produced. When the mutation is in the gene, it may cause the protein to be particularly hyperactive or resist being degraded and therefore last longer in the cell and push the cell around the cell cycle faster. Now, one of the best known oncogenes is called RAS. Under normal circumstances, RAS is a G protein that conveys a signal from a cell surface receptor through a series of protein kinases and eventually to the nucleus. A transcription factor in the nucleus causes the transcription of the RAS protein, which then stimulates the cell cycle to continue. Mutations in the RAS gene, however, enable the RAS protein to send the signal to the nucleus, even if there's no external signal received. That is, the RAS protein sends signals to the nucleus, and the nucleus generates a protein that causes the cell cycle to continue. A second type of gene is called a tumor suppressor gene. These genes serve like brakes on a cell, preventing the cell from progressing around the cell cycle. When tumor suppressor genes get mutated, it's like the brakes failing, and the progression goes around the cell cycle it's continuing without breaks. In fact, our genome and genomes of other long-lived organisms may have many tumor suppressor cell genes. However, given our long life, we incur more and more mutations throughout our life. And if these mutations occur in the tumor suppressor genes, then eventually the braking power of those genes is reduced, and we may develop cancer. Now, p53 is a well-known tumor suppressor gene that plays a wide variety of roles. When there's damage to DNA, this is identified and a signal is sent which activates the p53 protein, which is a specific transcription factor. As a transcription factor, p53 increases the rate of transcription of a protein that ultimately inhibits the cell cycle. However, p53, if p53 is mutated, then the inhibitory protein is never produced and the cell cycle continues. In fact, p53 plays a variety of roles. It can turn on expression of p21, which binds to cyclin-dependent kinases. It can reduce the possibility of entering mitosis. Additionally, p53 can turn on genes involved in DNA repair, and it can induce apoptosis by activating necessary genes when repairing the DNA isn't successful. Cancer usually progresses as a series of steps. Because our cells have many safeguards against runaway cell cycle, multiple mutations are necessary to cause most cancers. Loss of several tumor suppressor genes and mutations that convert proto-oncogenes to oncogenes will cause a progressively faster cell cycle in the affected cells. As the cells grow and reproduce, they often become sort of sloppy with their reproduction, and DNA synthesis causing more and more mutations. When there are enough accumulated mutations, cell growth runs out of control, and a tumor may spread from one part of the body to another, usually through the lymph nodes. When this happens, cutting cancer becomes much more difficult.